Okay, we are back for week 3. This is going to be a fairly easy week because we already covered almost all of the things on our outline for week 3 during week 2. It was sort of a repeat, give ourselves a little extra time. We've already talked about how DNA is made. We've already talked about the structure of chromosome. We've already talked about aneuploidy, basic patterns of inheritance were all covered. So now we're just going to finish up with X inactivation and chimerism. And then we'll do some case studies where we put this information into a clinical context so you can see how it looks in real life and start to think about the nurse's role, which is more next week's content, the professional role and what our responsibilities are in terms of knowing and applying this information. But we're going to keep going. So one of our last topics is X inactivation. Every biological female is sort of a mosaic. Not really. So if you remember our discussion of mosaicism, what you have in early embryonic development is a mistake in the duplication and reproduction of those chromosomes. When those chromosomes are dividing, maybe we get an extra chromosome, and so we have a mosaic trisomy. Some cells with the mistake will continue to reproduce that mistake and express the characteristics of a trisomy, whereas other cells will have the original genome that came from the maternal and the paternal chromosomes that were contained in the fertilized egg, the zygote, right? X inactivation is how some cells equalize the gene dosage between males and females. So here I have a slide. Equality between the sexes starts with the chromosomes. You'll notice that the X chromosome here on the left has a lot of genetic material. About 900 genes are located on the X chromosome. Females have 2 of these. Males, on the other hand, have an XY. The Y chromosome doesn't have that many genes. It has far fewer. So according to genome.gov, that number is about 55, although if you do a search of how many genes are on the Y chromosome, you will get numbers ranging from 40 to about 100. If anything, I hope that this course is teaching you that the recipe for a human being is complicated enough. You can't just add 900 ingredients and still get a normal human, right? That's the whole idea behind why trisomies can be so devastating. Most of them end in miscarriage early. So we have to equalize things somehow. We cannot just walk around with 900 extra genes coding proteins, that men don't have. In females, one X chromosome in each cell is inactivated during embryonic development, and it's turned into, let's get my pen out, make it purple. This here is called a bar body. When you look at female cells under a microscope, the nucleus will have this little dark spot. That is like crumpled up genetic material found on the X chromosome that was inactivated. So the process is considered to be random, and the outcome can be different for any given somatic cell. It happens in each cell independently of the other cells. So it's not like one X chromosome is chosen for everybody. It is chosen in each cell. It happens early enough in embryonic development that it will give rise to many other cells. So you'll see the same pattern in, a lot of different tissue types and things like that. Some of the genes on the inactive X escape. They're like the rebels and poets, and they're going to express themselves. They don't care that you crumple them up into a ball. I want you to sort of think of this like screenwriters in that movie script that we were talking about. They looked at a script from 2 different angles and decided which one to go with in the version of that film. The cell does kind of the same thing. And maybe a few lines from the extra scene are preserved and they make it into the filming of the movie. But most of it is edited out. It's sort of trashed. It's something that they don't use. The end result means that we have an approximately equal number of genes expressed in both males and females. And that is what X inactivation is, and it's an important thing. So some questions you might have. I tried to think like a student when I made this list. Is X inactivation always random? Well, it is thought to be. There are some articles. If you're curious enough, you can look them up. I certainly would not test on that. About skewed X inactivation, where sometimes we see those X-linked recessive disorders in a female with a relatively normal genotype. They have the dominant allele, but for some reason, the X chromosome that was selected to express all of its traits was the one with the recessive allele. It happens. It's just not very common, skewed X inactivation, and it is thought to be a random process. Does X inactivation change the genotype or just the phenotype? Well, that really could be a test question because the genotype is still there. All of that DNA is still in each and every cell, even though every cell will express that differently. So the phenotype could be different because not all of the genetic material is expressing itself, but the genotype is the same. We'll talk more about identical twins and clones because that kind of illustrates the concept. It's a couple questions down. Anyway, if one X chromosome is inactivated in every cell, why aren't there more biological females affected by X-linked recessive disorders like colorblindness? I was really curious about this. Don't you need all of the proteins that those don't you need all of your X chromosomes to express that dominant trait to have normal color blindness, normal color vision? It turns out you don't. You only need about 50% of the rods that X chromosome will code for. You will still have normal color vision without all of those alleles expressing their traits. However, sometimes you get females with color blindness and they are heterozygous. And that can happen because they have fewer than that 50%. Just randomly, those were the cells that selected that recessive allele or that recessive allele on that X chromosome that was selected. And so they have reduced color vision, but they still have some differentiation of red and green. Has happened. But most of the time, you only need to express the trait 50% of the time to have normal color vision. Why do females with Turner syndrome have symptoms? You only need one X chromosome. Well, that is why Turner syndrome is the only monosomy that is compatible with life, because only a very small amount of genetic material is missing. Only the material that's found on those escape genes. Those genes regulate things like skeletal growth, and they regulate secondary sex characteristics in females. So when you inactivate the X, you still have a few genes that express traits, those escape genes. The female with Turner syndrome does not have those escape genes, and so they are shorter in stature, and they have some differences in the female sex characteristics. They don't go through puberty. They have a uterus, they have ovaries, but they are non-functional. They cannot reproduce normally. They don't have enough genetic material to fully express the female secondary sex characteristics. Are identical twin females really identical? Yes, they have the same genotype. However, occasionally you will find identical twin females with some subtle differences, particularly in things like height, sometimes in psychiatric disorders, and it is thought that that is due to X inactivation. So phenotypically, they could be very subtly different. Remember, it's just the X chromosome that gets inactivated. You have 22 other pairs of chromosomes, and they will all be expressed consistently between those two females. However, some small differences may exist based on X inactivation, because at random, their cells chose one over the other in every cell. What about triple X or Superwoman syndrome? Why are they usually phenotypically normal? Well, because the exist center, that's what it's called, didn't make it up, will select, it counts the chromosomes, and if there's extra X chromosomes, it will inactivate both. So usually with Superwoman syndrome, which is 3 X chromosomes, it is a 47XXX female. You get a woman who has normal intelligence, normal facial characteristics. Sometimes there's differences in height, but they are capable of bearing children, and that is due to X inactivation. Some women don't even know they have it until they're karyotyped for some reason. Maybe they're going through fertility treatment and someone does a karyotype, or there's some familial syndrome and they need that. So usually they're phenotypically normal. They are capable of bearing offspring because only the X chromosome that was selected is expressing its traits, except for the presence of a few escape genes. Klinefelter syndrome, or XXY. It is a trisomy. Not many trisomies are compatible with life, but Klinefelter's is because that second X chromosome on this biologically male individual is inactivated. With Klinefelter syndrome, however, we do see changes in the male secondary sex characteristics. Generally speaking, there is reduced hair growth, body hair growth. We see smaller testicle size, higher voices, things that are associated with the normal level of testosterone. People with Klinefelter syndrome tend to be deficient in testosterone, and that is because of those escape genes exerting their influence inappropriately. Just a little bit of extra genetic material. So now you understand you can't add 900 genes to a human and get something normal. In fact, just this presence of a tiny amount of extra genetic material can make a big difference in the way that person presents, their phenotype, and also functions. Okay, chimerism, last concept, and it's not even in your book. I am not testing on it, so if you want to cut this video off here, go ahead, but it's kind of interesting, so I wouldn't. It only takes a minute. A chimera is a mythical creature composed of 3 beasts in one body, or more than one beast in one body. So we have like the ram, we have a lion, we have a serpent, and they are all sort of connected with this one body. What chimerism is, it is having some cells with the original genotype and some cells with the genome of another individual. That other individual's genome is expressing its traits. Most commonly, we see this with stem cell transplants. So if we start to talk about cancer and we talk about stem cell transplants, what we're doing is we're giving somebody a dose of a pluripotent, remember that word, it's a cell that can become anything it wants to be. It's a blank slate, but it has all of the genetic material of the donor. It has the exact same blueprint, the same copy of the script. Different movie, though. When we transplant stem cells, their function is to create new cells when the host can't do that for themselves. So if you have severe anemia caused by chemotherapy, or you have bone marrow that won't produce white blood cells, we give them this transplant of somebody else's stem cells. This is when we have donor transplants. If you have autologous, that's injecting somebody with their own stem cells, completely different. When we see chimerism in patients who have had stem cell transplants, we know that it worked. We know that the donor cells went ahead and made new cells. They did their job. But it can make sequencing the original genome for that person to, really target their condition with medications that address either a genetic issue or medications that their body's going to tolerate better and use more effectively. It can make that process a little trickier because now you have 2 genomes in one person. Microchimerism occurs. It's a smaller amount of chimerism. The most common cause of microchimerism is having a baby. So women who have given birth sometimes have fetal cells circulating in their body or in their tissues, situated in their tissues. This clinical significance of this is sort of not well understood yet, but there is a possible connection to autoimmune disorders because you have a foreign DNA in your system that may be coding for some proteins that are different from proteins your body recognizes as normal. And so, autoimmune disorders, which are known to be more common in women and can be aggravated after a woman gives birth, things like lupus, multiple sclerosis, more common. There is some literature that suggests that the more children you've had, the higher your risk for autoimmune disorders like that is. However, it's not really conclusive, and other articles seem to suggest that this is not a bigger player. It's not clinically significant. So stay posted for more information on that. Again, not testing on it, but something you might want to look at. Some cases are reported in recipients of blood transfusions or organ transplants, and that's why most blood centers are now releasing leukocyte reduced blood, because it tends, when you have somebody who needs a lot of blood transfusions, for example, sickle cell patients, one of the big therapies, it's a chronic disease, they're going to have it all their lives. They have multiple blood transfusions over the course of a lifespan, they can develop antibodies. So it's, they're more likely to have a transfusion reaction. They're more likely to suffer from autoimmune complications. So some cases of microchimerism have been reported in recipients of multiple blood transfusions and organ transplants. That DNA sometimes, of course, the organ is going to have all of the genetic material of the donor, but sometimes you see it in the blood, things like that. That is the end of our discussion for today. We're going to come back with some case studies to help you apply this material and put it into a more clinical context. It won't be as dry. And okay, I'll see you in a little bit.